So, hi everyone. My name is Shakel. I'm a PhD student in Tamir Tuller's lab, and I'd like to talk to you about my project and how we're improving the prediction of CRISPR efficiency using the 3D structure of the genome. So, you've probably heard of CRISPR before. It's a very important tool in biology. It lets us edit DNA in living cells and organisms. And the basic way it works is here you have the CRISPR complex, which is made up of a nuclease called Cas9 and a guide RNA. And this guide RNA guides the complex to a complementary target site in DNA. And then upon binding to that site, Cas9 induces a double-stranded break. And this break usually leads to a mutation, usually either an insertion or a deletion. So CRISPR is a way to edit cells, to edit DNA in living cells. But it has many problems. For example, in many cases, it doesn't edit where we want it to edit, so it doesn't affect the on target, but it does affect some other undesired regions of DNA, so it affects off targets. And so while CRISPR is full of potential for scientific and medical uses, this potential cannot be fulfilled yet because we cannot use it precisely. Right now, the use is relatively imprecise. And for that, to that end, several computational models were developed in the last decade. Computational models for the prediction of CRISPR efficiency, the probability of inducing a mutation, of editing the genome, but there's still a lot of room for improvement. And the main reason is that we don't fully understand what affects CRISPR, what are its determinants, and how to predict the mechanism and its outcomes. So what do we know? Well, the current models mainly focus on local features that have to do with the target site itself, with the flanking sequence, and the sequence of the target site itself. And so we have GC content, sequence motifs, mismatches and bulges between the gRNA and the target site, and in some cases, the biochemical properties of the target sites, such as the melting temperature and epigenetics. And here, we're proposing a new type of feature, which wasn't used before, and this is the 3D structure of the genome, so the way DNA is folded in the 3D cell. And we aim to use that feature in order to improve the prediction of CRISPR efficiency, as well as gRNA design and the use of CRISPR in general. So let's see how we did it. Well, first, we needed two types of data. First, 3D data in order to generate the features, and empirical measurements of CRISPR efficiency to evaluate our features. And I'll go over each of these one by one, beginning with the 3D data. So this is the general flow of our data generation. We began with a high-C interaction matrix. We converted it into a weighted graph. And based on that graph, we estimated the pairwise 3D distance between chromosomal regions. So as I said, we began with a high-C interaction matrix. This was published in 2020 and generated from T cells in 10,000 nucleotide resolution. And so each chromosome was divided into 10,000 nucleotide regions. And here we have quantifications of the interactions between these regions. And so based on this matrix, we created a weighted graph where each node is a chromosomal region. Again, resolution of 10,000 nucleotides, which is a high resolution in high C. So each node is a region, and each edge between nodes is the distance between them, where we assume that distance is inversely proportional to interaction, since regions that are far away from one another will interact less. So we created this graph, but this wasn't enough, since high C data is in general noisy and usually doesn't represent the full picture of interactions in the chromosome. And to that end, we wanted to acquire a more robust estimation of the distance. And so we did it in the following way. So let's say that we're considering this pair of chromosomal regions, nodes in our graph. We looked at all the possible paths between them, and we took the p shortest paths. In this example, we took the 5 shortest paths, but this is, in general, the results were very, very similar for different cutoffs. So we take the 5 shortest paths between these nodes, and the distance would be the average of these paths. This way, we acquire, as I said, a more robust estimation of the distance between them. And in this specific case, as you can see, there even isn't a direct edge between these nodes. That doesn't mean that they don't interact. That simply means that this interaction wasn't captured by high C. And so here we're trying to alleviate that a little bit. So this is our 3D data. Again, we began from high C and estimated the pairwise distance matrix in each chromosome. Okay, so this is our 3D data. Next, we needed CRISPR efficiency data to evaluate our features. And this is the data set we used, published in 2019. And it has two main advantages for us. First of all, it is endogenous. Rather than being measured in vitro or in artificial target sites in plasmids, this is endogenous, and it's important since we're considering the target sites in their natural context in the 3D cell. So this was important for us. Second, this data set also includes the genomic location of each target site, rather than only the sequence, which is what most data sets include. This is important for us as well, since the location of the site is the basis for the generation of our features, the feature which is based on the location in 3D. So this is the data set we used, and the general way it was produced was by electroporating T cells, and then following the CRISPR cleavage, the on-targets were mapped, and then the efficiency was quantified based on the repair profile at the targets. And so this is what our efficiency data looks like. We have a list of sites, each one with a genomic location, and an efficiency score between 0 and 1. This is simply the probability of editing the DNA. Okay, so this is our second data set, the CRISPR efficiency. And it's important to note that both of these are taken from the same cell type. It's important since both the 3D structure of the genome and CRISPR efficiency can vary between cell types. And so it was important for us to have data from the same cell type, although these are completely different experiments, different labs. The 3D data originally has nothing to do with CRISPR. So they're not related in any way, except for the fact that they're taken from T cells. So this is our data. And let's see what we did with it. So we wanted to estimate the local density of each chromosome. And we wanted to move from a pairwise distance matrix to a region-wise or node-wise density measure. So this is the way we did that. So let's say if we're considering this particular region right now, we want to estimate its density. And so we're considering this region, and we're looking at all the closest points in a distance matrix, all the closest neighbors, and we tried a few different cutoffs, as you can see here. So we're taking the n closest points, and then we're simply taking the average of their distances. So we're taking all these neighbors, and and the feature would be the average of all these distances between our node of interest and its neighbors. This is the feature. And this is actually a measure of sparseness in the chromosome, since a high average distance, a high feature means that the closest neighbors are actually relatively far away. And so in this case, this is a measure of sparseness, but it's the same idea. And as I said, we tried a few different versions and cutoffs of this feature, but from here on out, we'll only talk about a specific version of this feature where we take the closest 2,500 neighbors. So this is our 3D feature. And in order to evaluate it, we compared it to 5 state-of-the-art models. These are CRISPR efficiency models that are very highly regarded and have a very high performance relative to other models. And as you can see, 2 are linear regression models, 2 are deep learning, and 1 is SVM. So these models would be the benchmark for our feature. And when we evaluated our 3D feature, we had 3 main questions. First, does our feature perform comparably to these models? Second, do we add new information that is not encoded in these models? And third, do we improve our ability to predict CRISPR efficiency? And I'll tell you ahead of time that the answer to all these questions is yes, but let's see that in action. So we began by looking at the correlation between the 3D feature and CRISPR efficiency. As you can see, the correlation between the correlation of the 3D feature is comparable to the 5 predictive models, which I talked about earlier, even outperforming 3 of them. And this is especially impressive when you consider that each of these models contains multiple features and was trained separately. And this is a single feature that was not trained on anything, and as I said, taken from a data set that has nothing to do with CRISPR. And still it is comparable to these models. So these are very encouraging results regarding our feature information. So that's the first point. Secondly, we found a consistent trend among all the different versions of feature we created, where the 3D density of the genome was inversely related to CRISPR efficiency. This may be due to inaccessibility of these regions, but this is a relation that we intend to investigate in the future. You can also see that here, since, as I said earlier, this feature is a sparseness feature, the green one, and it is positively correlated with efficiency. And so dense regions are less efficient and vice versa. So this is the second point I wanted to talk about, where this trend we found, the inverse relation between 3D density of the DNA and CRISPR efficiency. Next, we looked at the partial correlation with CRISPR efficiency when controlling for the other models or features. So as you can see, the partial correlations of the existing models is significantly lower. The correlations are significantly lower than the regular correlations, meaning that they encode very similar information to one another. Whereas the partial correlation of the 3D feature, even when controlling for the 5 existing models, is largely the same, meaning that it indeed adds new information not encoded in these models. And this actually makes sense, since, as I said in the beginning, the current models mostly look at the local features, at the sequence of the target site, and here we're adding completely different data that cannot be explained by the sequence of the target site itself. And so to answer our first 2 questions, yes, our feature does perform comparably to the existing models. And yes, it does add new information that cannot be explained by them. Next, we wanted to see if we can improve the predictive power of CRISPR efficiency. And we did that in the following way. So we trained two linear regression supermodels, where each supermodel includes the existing models I talked about. And so the first supermodel simply includes as features these 5 models, and the features are just the predictions of these models. This is the first supermodel. And for the second one, we added a 3D feature, but it is otherwise the same. And we wanted to see if the second supermodel is significantly better than the first one. And so to do that, we conducted a process which is similar to bootstrapping. We took a random 80% of the CRISPR efficiency data, which we talked about earlier. We took a random 80% and trained both of the supermodels on it. And then we tested them on the remaining 20% of the data set. And so we acquired a performance, a correlation between the measured and predicted efficiency. We actually acquired two correlations, one with and one without the 3D feature. We repeated this process 1,000 times, each time taking a random 80% training set and testing on the remaining 20%. And so for each iteration, we received 2 correlations, one with and one without the 3D. And so what we acquired in the end are 2 distributions, one, the performance, the correlation of the model with the 3D, and a distribution of 1,000 correlations for the model without the 3D. And here you can see the results. So these are histograms of the correlations. The blue bars are the model without the 3D. The green bars are the model with the 3D. And as you can see, adding the 3D model indeed improved the performance. In fact, the performance was improved significantly. So the R squared was increased by 24%, and the difference between these distributions is significant. And so indeed, adding the 3D features improved the predictive power of these models. Lastly, we wanted to approach this from a slightly different angle and look at feature selection. And so we repeated a process similar to what we described earlier with 1,000 iterations, but we used either stepwise regression or lasso to conduct feature selection and count the number of times each feature was selected. As you can see, the 3D feature was selected in all 1,000 iterations, while the other models had varying results. Again, this demonstrates the importance of the 3D feature, and the improvement in predictions was significant here as well. As you can see, an increase of 28 or 25% in the R squared. And so to answer a third question, yes, we do improve the predictive power by adding the 3D feature. So to sum up, as I've shown you, the 3D density feature is indeed informative. It is correlative with CRISPR efficiency and adds new information which is not explained by current models. I've also talked about this consistent trend we found where the 3D density was inversely proportional to the efficiency, and I suggested that this may be due to the inaccessibility of these regions, but we still intend to investigate this further and in other cell types as well. And lastly, I've shown you how adding the 3D feature significantly improved our ability to predict CRISPR efficiency. And this leads me to a more general conclusion where the global cell-based features should be considered in addition to the local features. So as I said in the beginning, the current models, the focal point of the current models are the local features, the target site, and this is very important, of course. But as we show here, considering in addition to the target site itself, the properties of the cell can help us significantly in improving the prediction of CRISPR efficiency and the usage of CRISPR in general for scientific and medical uses. I'd like to thank my lab members. I'm a Software Center Fellow at Tel Aviv University, and this project was part of the CRISPR Consortium. And thank you for your attention. I can't see or hear you or anything, so.
From the audience live. Let's see if you can hear them.
Hey.
Yeah.
Hey, thank you so much. Great talk. Can you hear me?
Do you hear
Yeah,
me?
I can hear you.
Yeah, great talk. Thank you so much. So the 5 state-of-the-art models that you mentioned, do they output features or do they output CRISPR efficiency predictions?
So, you're asking about the 3D feature. The sound is a bit muffled. You're asking about
the 5 state-of-the-art models that you mentioned that within your
work.
Is the output some features or is the output the efficiency prediction?
Okay, so the output is the efficiency prediction. They're trained on multiple models, but what they are predicting is the CRISPR efficiency. This is what I checked. So yeah, so they're based on multiple features. As I said, I hope you can hear me with all this echo. I hear some terrible echo. These models are based on various features, but what they took is simply their prediction for efficiency, and this is what they indeed predict, the efficiency of CRISPR.
I think, thank you. So, Mac, then reaching to my question, what I'm trying to say is like you build these linear super models and you're sort of doing this linear regression, but the outputs of the 5 models are CRISPR efficiencies and you're adding to that a feature. So I found that a little weird and hard to digest. Could you explain that a little bit? Does that make sense? Because like 5 of them are efficiency scores and one is a 3D density feature that you have come up with.
So yeah, I will explain, of course. The basic assumption we had. Sorry, I have some terrible echo. I'll try maybe to lower the volume at my end. The basic assumption we had is that if adding the 3D feature wouldn't be really informative, then simply training these supermodels. I'll just go back to that slide so you can look at it. If the 3D feature doesn't add new information and cannot improve the prediction, then I would expect both of these supermodels to do very similarly, since they directly predict the efficiency of CRISPR, then they should be good enough on their own. And so I agree that we're adding here some feature which is not directly the CRISPR efficiency, but since it is correlative with it, then it can be used as a feature in that model. So our intention here was only to show that adding this feature can indeed improve the predictive efficiency, the power of prediction. We didn't aim here to train a whole new model. We didn't want to create a new model of CRISPR efficiency. We only wanted to check this specific feature. Since the output of these models should be sufficient on its own. But as you see, it's not, and we think that it means that in addition to, as I said at the end, in addition to just considering the target sequence, you should also consider the cell-based features, the properties of the cell. Again, if I think if the 3D feature wasn't significant, then I would expect both of these supermodels to do roughly the same. Since the prediction is already the prediction of the 5 state-of-the-art models should be mature enough on its own. But as you can see, it's not, since it's missing a crucial part of information regarding CRISPR.
Okay. Hi, thank you. That was very interesting. Does this relate to the AB compartments that have been inferred from high-C, or in general, like open and closed from high-C?
I'm sorry, can you repeat the question? I couldn't really hear you.
Yeah. Did you check whether this corresponds to the so-called AB compartments, meaning generally open or closed chromatics?
I still cannot understand this question. I'm sorry. Could anyone write in the chat, maybe?
Hi, the question is, does this relate to AB compartments in chromatin?
Okay, I understand. We didn't check directly about AB compartments, since AB compartments are generally very low resolution structures, and here we wanted to look at it from a very high resolution scope. As I said, 10,000 nucleotides. This is very high resolution regarding for high-C. AB compartments, as far as I know, are usually like 100,000 or a million base pairs. So we didn't check it. We didn't check the AB compartment angle, mainly because it's too low resolution. But it is, of course, a very interesting direction to check the information of high-C. But we wanted to be a bit more granular and a bit more high resolution than that. So we didn't check it yet, to answer your question.